Every day, the human body is challenged by invading microbes. Whether an infection is cleared or a disease develops depends on the dynamic interactions between host immune defenses and microbial virulence strategies. The immune system must constantly ask, are innate and adaptive mechanisms sufficient to stop the invasion? Microbes come in many forms: bacteria, viruses, fungi, and parasites. Each category possesses unique structural and metabolic features and, therefore, stimulates a distinct immune response. Importantly, microbes are not passive targets, they continuously adapt to immune pressure. A key concept in immunology is the classification of pathogens based on their location. Microbes may exist extracellularly, outside host cells, or intracellularly, hidden within host cells. This distinction is critical because it determines which immune mechanisms will be effective. Extracellular microbes remain accessible to antibodies, complement proteins, and phagocytic cells such as neutrophils and macrophages. Because they are exposed, they are ideal targets for opsonization and phagocytosis. The first line of defense against extracellular microbes is innate immunity. Neutrophils rapidly ingest microbes, while complement is activated mainly through the alternative and lectin pathways. Complement fragment C3b mediates opsonization, while C5a is a potent inflammatory mediator. Acute phase proteins, such as C-reactive protein, further enhance opsonization. Adaptive immunity reinforces innate defenses through antibody production. Immunoglobulin G antibodies opsonize microbes, activate the classical complement pathway, neutralize microbial toxins, and block microbial attachment to host cells. Certain thin-walled gram-negative bacteria, such as Neisseria, are directly lysed by the membrane attack complex formed by complement components C5 through C9. Clinically, deficiencies in terminal complement components are associated with recurrent Neisseria infections. Some extracellular pathogens cannot be effectively cleared by antibodies alone. In these cases, cytokine-mediated neutrophil recruitment is essential. This is where T helper 17 cells play a central role. T helper 17 cells secrete interleukins 17 and 22. Interleukin 17 promotes neutrophil recruitment, while interleukin 22 enhances epithelial barrier integrity and antimicrobial peptide production. Together, these cytokines strengthen defense at mucosal and epithelial surfaces. T helper 17 responses are particularly important for protection against extracellular fungi, such as Candida and Aspergillus. Defects in T helper 17 immunity lead to chronic mucocutaneous candidiasis. In contrast to extracellular microbes, intracellular pathogens reside within host cells, effectively hiding from antibodies and complement. As a result, humoral immunity alone is insufficient for their elimination. Natural killer cells provide early defense against intracellular pathogens. They kill infected cells, especially those with reduced MHC class I expression, and produce interferon gamma. However, innate immunity alone cannot fully eradicate intracellular infections. Clearance of intracellular microbes depends on cell-mediated immunity. CD8 cytotoxic T lymphocytes induce apoptosis of infected cells, while CD4 T helper 1 cells activate macrophages through interferon gamma, enhancing intracellular killing. Parasites, particularly helminths, pose a unique challenge to the immune system. Before adaptive immunity develops, the innate immune system initiates early defense against helminths. However, unlike bacteria or viruses, helminths are large multicellular organisms that are too big to be phagocytosed by macrophages or neutrophils. As a result, classical phagocytic mechanisms are ineffective, and the immune system must rely on alternative strategies to eliminate these pathogens. Defense against helminths is dominated by T helper 2 mediated immunity. When antigen-presenting cells encounter parasitic antigens, they drive CD4 T cells to differentiate into T helper 2 cells. These cells secrete key cytokines, interleukins 4, 5, and 13, which collectively coordinate the anti-helminth immune response. One of the most important effector cells in parasitic infections is the eosinophil. Interleukin 5, produced by T helper 2 cells, stimulates eosinophil growth, activation, and survival. Parasites become coated with immunoglobulin E antibodies, and eosinophils bind to these antibodies through Fc receptors, releasing toxic granules that damage and kill the parasite. This process is known as antibody-dependent cell-mediated cytotoxicity, or ADCC. In addition to eosinophils, mast cells play a crucial role in eliminating helminths, particularly at mucosal surfaces such as the intestine. When immunoglobulin E on mast cells binds parasitic antigens, mast cells degranulate, releasing mediators that increase mucus secretion and enhance smooth muscle contraction. These effects help physically expel helminths from the gut, preventing long-term colonization. Nevertheless, microbes evolve multiple immune evasion strategies to survive. These mechanisms allow persistence, chronic infection, or repeated reinfection despite an active immune response. Let's see how the sneaky microbes escape our immune defenses. We'll start with antigenic variation, which involves frequent changes in microbial surface antigens, preventing effective antibody binding and immune memory. Influenza virus antigenic drift is a classic example, which involves the gradual continuous accumulation of point mutations in the genes encoding the surface proteins of the influenza virus. Some microbes interfere with antigen presentation by inhibiting MHC class I or class II expression. This prevents T cell recognition. Cytomegalovirus and other herpesviruses use this strategy. Encapsulated bacteria are protected by a polysaccharide capsule that acts as a slippery shield, preventing effective attachment of phagocytes. This capsule also masks microbial surface antigens, making direct phagocytosis inefficient. As a result, immunoglobulin G antibodies and complement component C3b become essential. Immunoglobulin G binds to the capsule and engages Fc receptors on neutrophils and macrophages, while C3b binds to complement receptors, together enabling effective opsonization and phagocytosis. Surprisingly, some microbes survive even after being engulfed by phagocytes, resisting intracellular killing mechanisms. Mycobacterium tuberculosis is an acid-fast bacillus that can prevent fusion of the phagosome with the lysosome, allowing intracellular survival. Clinically, defects in the interleukin-12 T helper 1 interferon gamma axis lead to severe, disseminated tuberculosis. Other bacteria, such as Salmonella typhi, can survive within the phagolysosome by resisting lysosomal enzymes and oxidative killing. Inside the phagosome, Listeria monocytogenes refuses to stay trapped. Using its Listeriolysine O, it breaches the phagosomal membrane and escapes directly into the cytosol. Living freely in the cytosol means its antigens are processed via the MHC class I pathway. This would normally trigger CD8 cytotoxic T cells. However, the bacterium hijacks the host's actin. Actin filaments rapidly assemble at one pole, forming a blazing actin rocket tail. Propelled at high speed, Listeria shoots through the cytosol and even pushes into neighboring cells, cell to cell, without ever re-entering the extracellular space. By spreading directly between cells, Listeria partially evades immune detection. Some microbes evade complement by expressing surface molecules that inhibit complement activation or accelerate complement degradation. Neisseria meningitidis is a key example. Certain microbes actively suppress host immunity by inhibiting cytokine production or inducing immune cell apoptosis. Examples include human immunodeficiency virus and Epstein-Barr virus, which produce viral interleukin-10 homologs. Loss of CD4 T cells impairs macrophage activation, disrupts cytotoxic T cell responses, and reduces antibody production. This explains the susceptibility to opportunistic infections seen in acquired immunodeficiency syndrome in the late stages of HIV infection. The last example of immune evasion mechanisms today is biofilms. Biofilms are structured microbial communities that protect organisms from antibodies, complement, and phagocytes. Common biofilm-forming organisms include Pseudomonas aeruginosa and Candida species. Biofilms promote persistent, recurrent, and treatment-resistant infections, making eradication particularly challenging in clinical practice. In conclusion, effective immunity depends on matching the immune response to the pathogen's location and strategy. Understanding whether a microbe is extracellular or intracellular allows us to predict the dominant immune mechanism required for protection.